Alright, hi everyone. I hope you guys are doing well. So this lecture video is going to be on chapter 17 about learning and memory. So this is the video for the week of April 6th. We do have activity 11 that's going to be due by 5 p.m. on April 10th, so next Friday. However, remember that you do get to drop grades for activities. So if you have gotten credit for the first 10 activities, then there's no reason as far as your grade is concerned that you need to do activities 11 and 12. However, if you have done activities 1 through 10 and you just want to do activities 11 and 12, that's great. But if you have missed an activity or two from earlier in the semester, then I would advise you to use your drop grades there and to complete activities 11 and 12. But let me know if you have any questions about that. Also, looking ahead, we're going to have exam 4 that's going to open on April 13th, and it's going to be open until the end of the final exam period. So this is the schedule for the 8 o'clock class, so the final exam period ends for them at 11 a.m. on April 24th, and then for my later class for section 2, which is at 11, right? I've already been out for a couple weeks and forgotten my schedule, but for my later section, my 11 o'clock, yeah, it's 11 o'clock, that's going to be open until the end of their final exam session, which would be at 3 p.m. on April 29th. Okay, so let me know if you have any questions. I will be posting a study guide for that soon, and I will also do a review video for exam 4, just like I did for exam 3. Alright, so as I said, today's video is going to be on learning and memory. So when we talk about learning and memory, we might think about them as being the same thing. They definitely do go together. Learning, one way of thinking about this could be the process of acquiring new information, which you might think about in a class. You're learning the information that a professor is teaching, but this starts at really birth. We learn the language that is spoken to us. We learn the customs, we learn the ways of dressing, the ways of speaking that we hear and we see. So we are learning all the time throughout life. However, you might ask yourself the question, how do we know that learning has happened? For example, how does a professor know that his or her students have learned something in class? We test your memory for that. So memory is it could be defined a couple of different ways. You could think of it as the ability to store and retrieve information. So this is an ability, versus you could think about a memory as being a specific piece of information that is hopefully stored in your brain, that hopefully when you go to find it, it's there where you are easily able to access it. Now, this has been a theme throughout this class, that we have studied how the brain works by finding individuals that have had accidents or surgeries, they have some type of brain damage, and then seeing what kinds of symptoms they have. So this is the case for memory as well. A lot of what we know about how the brain works with regards to memory, we know because of case studies, so specific individuals that had issues that we've studied. Now, here you see patient HM. You'll see the initials being used a good bit for these case studies. If the person is still living, they use initials to keep that information anonymous, private. But Henry Molaison is no longer living. He passed away not too long ago. I can't remember what year it was that he passed away, but it hasn't been too long ago. So when I was taking undergrad psychology classes, he was still just called HM. But now we know that his name was Henry Molaison. So Henry Molaison had severe epilepsy that was not responding well to other treatments. And we've talked about throughout the semester how there are situations where if epilepsy is untreated and it's causing this severe dysfunction, or if we've tried to treat it and it's not working well, it is possible that we might go to some type of neurosurgery. So what happened here is that his seizures were beginning in the temporal lobes. So they decided that nothing else is working. Let's see if we can remove large portions of the temporal lobe and see if that stops the seizures. So, to a certain extent, if you're thinking about it that way, Henry's surgery was a huge success. His seizures stopped. The problem was he had some pretty significant side effects, as you might imagine. So what happened was that they removed the anterior or the forward temporal lobes on both sides of his brain, which included the amygdala, the hippocampus, and some of the cortex as well. Now, of course, when you remove a person's amygdala, there are issues there. You remove cortex, there's issues there. But specifically for this chapter, we're especially interested in the hippocampus. What happens when you have damage to the hippocampus or the hippocampus is removed? Well, in order to understand here what happened with HM, we have to clarify a couple of terms. So, amnesia is a general term for a major memory impairment. A couple of types that are mentioned here: retrograde amnesia. Retro is Latin for backwards. So we're talking about the loss of memories before whatever the event was. Now, this is not uncommon. Let's say, for example, you were in a car accident and you couldn't remember what happened right before the car accident. That's not unusual. Sometimes people might even have a loss of memory for days or weeks before the event. Maybe you have surgery and you lose some of what happened in the days leading up to the surgery. However, movies and books tend to get this wrong most of the time. You might have read books where they had a character, or seen a movie where there was a character that had complete retrograde amnesia, where they couldn't remember their own name. Like, who am I? They had no recollection of their past. That's not something that's ever been discovered as far as like research is concerned, but it's something that fiction plays up quite a bit. However, milder cases of retrograde amnesia are common. What is less common is anterograde amnesia. So with anterior, we're talking about Latin for like ahead, forward. So what's happening here is that an individual is losing the ability to make new memories. So maybe you had an accident or you had surgery and you were not able to form new memories. This does happen, but it is pretty rare. And what happened with HM is that he had severe anterograde amnesia. So he could remember most of his life before the surgery, but he was unable to form new long-term memories afterwards. Now, he had normal short-term memory. This, and we're going to talk about these terms later, but this tells us that there are different types of memory. If you can have a loss of the ability to form new long-term memories, but retain the ability to form new short-term memories, then we know that those are happening in different locations in the brain. So if you called out a list of words to HM and had him say them back to you right afterwards, he could do that as well as you can. But if you called out a list of words and then you distracted him with some other task, and then you said, now tell me about that list of words, not only would he not remember the words, he wouldn't remember that there was a list, right? He lost the ability to form new long-term memories, okay? Now, the thought is that the damage to his hippocampus, or really having his hippocampus removed, is what caused the problems for HM. Because there have been other situations where people have had damage to the amygdala and the cortex and have not had the severity of the symptoms that HM had. So it seems like his problem was with the hippocampus. HM has taught us something already, that long-term and short-term memory are happening in two different places in the brain. But he taught us something else as well. HM taught us the difference between more verbal information, or more of an information that you tell to another person, versus skill information. So HM was not able to gain new verbal memories that he could tell you about, but he was able to acquire new skills and improve his motor skills with practice. So there's a picture here of a person doing a mirror tracing task where you can't see the paper directly. You're having to watch the paper in the mirror and tracing that. I've never tried to do that, but I can imagine that would be challenging. So what happened is that in the beginning, HM was not very good at this, but they had him continue to practice it throughout the day, and then the next day they'd have him do it again, and the next day they'd have him do it again. And what they found was that he got better at this, but he didn't remember having done it. When the person said, have you ever done this mirror tracing task? He said, no, this is my first time. Well, he'd been doing it for days, so he couldn't remember the event of having done it before, but it's like he had the muscle memory, his motor skills improved. So this also tells us that the more skill-related memory is definitely stored in a different location than the more declarative memory, which we'll talk about in a second, but the memory you can tell someone about, right? Another example of a skill that people can learn is reading mirror reversed text. So I'm going to give you a second to try to read this. So I don't know how easy this task is for you. Of course, I've taught this class before. I've done this a couple times, but if you practice reading text that is mirror reverse, you will become better and better at, that's a difficult one, deciphering the text quickly. This is an example of learning a perceptual, does that say perceptual? That's tricky. Perceptual skill, and does not, okay, I'm already tired of this. Require an intact hippocampus. Okay, yeah. The idea here is that you can learn this skill of reading mirror reverse text if you practice it and practice it, even if you don't remember the earlier practice, even if you have damage to your hippocampus. So this tells us that there's a difference between declarative and non-declarative memory. So here we have this differentiation. Declarative memory is going to deal with what? So when you ask questions like, what did you do for your last birthday? Or what did you learn in biological psychology? Here we're asking for you to give information that you can tell verbally, right? You explain something. Now it's declarative because you're aware of learning it, you remember learning it, and you can express that through words, right? Non-declarative memory deals with how. So if you wanted to show me that you had learned something that was a skill, you would have to show me, not tell me. Like if I asked you to tell me how you juggle, I don't know how to juggle, so I couldn't, definitely. But tell me how you ride a bicycle. That's harder to do. You just show me how to ride a bicycle. So you might have a declarative memory of the first time you rode a bicycle, if you can remember that. But with non-declarative memory, you know how to ride a bicycle. You would tell me about your first experience with a bicycle, but you would show me how to ride a bicycle. And we know that these are two different types of memory that are stored in two different locations because someone who has lost their ability to form declarative memories can form non-declarative memories. So we've learned a lot of things from looking at individuals who've had damage, surgery, that kind of thing. Okay. Now, when we're talking about declarative memory, and we're talking about the memories that you can explain to another person, the answer to the what questions, there are two really flavors of this, two varieties of this here, recollection and familiarity, and they seem to happen in different locations. So perhaps somewhere in here there's a picture. If not, maybe I'll pull up a picture later and show you where I'm talking about here. But the perirhinal cortex is part of the temporal lobe. Well, I'll show you a picture later. But the perirhinal cortex is thought to be important for familiarity, while recollection is something that happens in the hippocampus. To give you an example to kind of tease these two apart, maybe, and I honestly, I haven't been inside a Walmart in almost a month, but maybe it's like you're in Walmart and you see someone and you think, oh, I know that person. Oh, they seem so familiar. Where do I know that person? And that nagging, I've seen this before, I've seen this person before, that's familiarity. But recollection comes when you say, oh yeah, that person was in my biological psychology class, that's why I know them. When you can recollect, you can remember the specific details of when you saw this person, where you saw this person. So depending on what part of the brain has damage, that might impact one or the other, or both if you have damage that's in both locations. All right, now let's think about another example of a person who's had an unfortunate experience that led to memory loss. So here we have patient NA. So the fact that your textbook gives only the initials tells me that this person is still living. They have not released this person's information, so we're just going to call this person NA. So NA suffered an unfortunate incident where, if I'm remembering correctly, this person was fencing. And the fencing, I'm sure there's a term for that. the sword that you use when you're fencing. I'm sure there's a technical term that I can't remember, and I don't have you guys here to help me remember things like that. But anyway, he was fencing, and the sword thingy went up his nose and caused brain damage. And he mostly recovered, but he had damage to his thalamus and hypothalamus. So remember that this is the diencephalon. Those two things together would be called the diencephalon. And thought to have damage to this mammalothalamic tract. That's a fun one. Say that one 3 times fast. Okay, what's happening here is that he's having damage to his thalamus and hypothalamus, and then also to his mammillary bodies. So I'll need to pull up a picture in a little bit to show you guys what I'm talking about here. But mammillary bodies are an important part of your memory system as well. What we're saying here is that his hippocampus was not damaged directly, but parts of the brain that connect to the hippocampus were damaged. So this leads us to believe that it's not just your hippocampus, that there is a circuit of different structures that are important for memory, and that if one or more of those structures is damaged, that will cause memory loss. So even though he did not have the specific hippocampus damage that HM had, he had similar symptoms. He had that profound anterior grade amnesia, so he lost the ability to form new memories. And so that tells us that there are lots of different structures that are all related to memory, and that damage to one or more of these structures is what causes the symptoms. Now, if we're talking about individuals who have memory loss, another important thing to note here would be Korsakoff syndrome. So Korsakoff syndrome is a health condition that happens when a person is lacking vitamin B1. So this is something that is often seen in chronic alcoholism. A person may neglect their diet and be primarily consuming alcoholic beverages. And so because of that, there can be a lot of nutrients that they're missing out on, and this thiamine in particular can be a problem. So there is some discussion about whether or not alcoholic beverages should be fortified with vitamin B1 to try to prevent cases of Korsakoff syndrome. But we could discuss that if we were in class, but I'll just leave that there. So what happens here is that there is damage to the individual's mammillary bodies and part of the thalamus, and that's very similar to what happened to NA. He had damage to his mammillary bodies and to parts of his thalamus and hypothalamus because of the fencing sword. But here we have an individual who has damage because of a lack of nutrition. In addition to the damage to the mammillary bodies and thalamus, though, he also, or I say he, I'm thinking about a particular case study of this, but individuals who have Korsakoff syndrome will also have damage to certain areas in their frontal cortex. Okay. Now, if you think about what your frontal cortex is important for, thinking, reasoning, decision-making, you'll see a lot of deficits that come along with Korsakoff syndrome. One thing that's unique to Korsakoff syndrome versus something like HM or NA is that people who have Korsakoff syndrome confabulate, which means that they make up stories. If you asked HM, what did you do this morning? How did you get to this room? What were you doing before you were here? HM would say, I don't know. I'm not sure. But someone who has Korsakoff syndrome might make up a story. And it might be a highly unlikely story, but they start making up a story that they seem to believe is true. They're not aware. It's not like they're lying to you, intentionally misleading you. They tend to believe their own stories that they make up. And the thought is that that's caused by damage to the frontal lobe that's not there in some of the other case studies that we talked about. So make sure you're eating a good diet. Take a vitamin. All right. So if we're talking about different subtypes of memory, we talked about how declarative memory is where you can explain something to a person. You can tell them how you learned it, when you learned it. But with non-declarative or procedural memory, you demonstrate to a person. You can ride a bicycle or juggle. Or I asked students once about what types of procedural skills they had, and someone said folding a fitted sheet, which, you know, is just super impressive. Okay, but now if we take declarative memory and we break it down into subcategories here, declarative memory is that memory that you can explain to someone else. Now, you could explain to someone else semantic information or episodic information. Semantic memory would refer to generalized memory, so knowledge of vocabulary words, one thing I did last semester was I asked students, what's the capital of Montana? And I had taught 2 sections of this class, and I don't remember how many students I had, but there was a good number of students. I only had 1 student that was able to remember the capital of Montana. Okay, so if you know the capital of Montana, feel good about yourself, okay? It's Helena. Anyway, but another thing that I did that I'm not doing this semester, just I would have done if we were in class, I would have done this, but we're not in class anymore. I would have students list the presidents, all the U.S. presidents they could remember in any particular order. I would do that to demonstrate something else that I'll talk about later, but also that's an example of semantic memory. That's just general factual information. Episodic memory is more specific to you, telling me about your first experience riding a bicycle, or what did you do on your last birthday. That would be episodic memory. So there's a patient, K.C. Your textbook gave his name, Kent Cochran, I believe. I think he recently passed away. He was injured in a motorcycle accident, so make sure you take your vitamin and, you know, be careful driving. But anyway, he was in a motorcycle accident, and he, in addition to some other problems as well, lost the ability to retrieve episodic memory. So he retained semantic memory, but lost episodic memory. So think about what that would be like. He would be able to talk to you about, you know, the capitals of different places, about presidents. Apparently he was very good at chess. He retained his ability to play chess, but he lost his personal memory. He lost the memories about his life, which, I mean, I think if I had to choose, I'd rather keep those, right? I'd rather keep those memories that are unique and special to me. So this is also showing us then that declarative memory is stored in different places depending on whether it's semantic or episodic memory, because here's an individual that maintained his semantic memory but lost his episodic memory. So it talks about he had damage to the cortex, so that could be something that might be somewhat similar to individuals who have Korsakoff syndrome, but then also having damage to the hippocampus and the parahippocampal cortex, so the cortex that is around his hippocampus. So you're going to be hearing about hippocampus a lot in this lecture. Okay. Now, if we were to break non-declarative memory down into different forms, non-declarative memory being the more memory that you demonstrate, not something that you can necessarily explain. Breaking that down into different categories, oftentimes when we think of non-declarative memory, we think of skill learning. Like I said, riding a bicycle, maybe playing a musical instrument, perhaps maybe shooting a basketball. I don't know, something like that could be skill learning. But then also priming. So it talks here about a change in stimulus processing due to prior exposure to the stimulus. So as an example, one thing they did with HM was they would have him read a list of words, and maybe one of those words was stamp. And then later, after he had forgotten that he had gone through that list of words, they would say, okay, I'm going to give you 3 letters, and I want you to make a word out of it. STA. What could you put at the end here to make that a word? And he would put MP. He would make it stamp. He would be more likely to use that word that he'd been exposed to earlier, even though he didn't remember being exposed to it. So that's non-declarative memory. And then associative learning, you'll see this, an example of this in a second. It's really getting at conditioning. The example in a second is going to be specifically classical conditioning, but the association of 2 stimuli or of a stimulus and a response. So I think we talked a little bit about this when we talked about fear conditioning in chapter 15. But what we're saying here is that you can be classically conditioned without having the ability to form new declarative memories. So you could have classically conditioned HM. So classical conditioning happens when you have an unconditioned stimulus naturally producing an unconditioned response, right? Nothing's happened yet. The food is an unconditioned stimulus that naturally causes the dog to salivate or to drool, right? That's an unconditioned response. Now, also initially, something like a bell is neutral. You ring the bell, the dog does not drool because there's no reason for the dog to drool yet. But then what we do is we take this neutral stimulus of the bell and we pair it with the unconditioned stimulus of the food. Ring the bell, give the food. Ring the bell, give the food. Over time, the dog will learn that association, and that bell will become a conditioned stimulus capable of producing a conditioned response. So now the dog will drool just because he heard a bell ring, because he learned the association between the bell and the food. If you have questions, let me know. That's just the classical example of classical conditioning with Pavlov. So long-term memory we've been talking about so far can be broken down into declarative, things you tell, which would be telling about yourself or telling just general knowledge, and things that you don't tell, you do, which you could show someone how to do a certain skill, or you might be more likely to choose a word you've heard recently, or you might be conditioned to salivate. You can condition a lot of things, not just salivation, really anything that's not a voluntary behavior. You can't condition someone to do a voluntary behavior, but you can condition someone to you can condition someone to be afraid of something. You can condition someone to panic, but you can also condition someone to feel happy, right? All these things. Behavior modification is an interesting class. I haven't taught in a while, but it was always one of my favorites to teach. Okay, now let's think about what's happening in the brain when we're learning new information, specifically learning information about locations. So it talks here about the fact that animals have a cognitive map. A lot of this research has been done in rodents, but then a lot of this has also been replicated in humans. So when we're talking about a cognitive map, we're talking about a mental representation of locations in your brain. So maybe you formed a map of your apartment or a map of your local grocery store, whatever the case may be. You have a cognitive map. That is your mental representation of spatial relationships. Now, when you take an animal and you place them in a new location, there are certain neurons in the brain that fire, these place cells in the hippocampus. Once again, the hippocampus is important for a lot of things related to memory, that fire when an animal is in a particular location. You have different cells, grid cells, neurons that are firing when an animal crosses intersection points. It's like your brain is constantly updating the map. It's like you have a GPS in your brain, and so whenever you come to a particular intersection where you could go right, you could go left, you could go straight, you could go back, these cells are firing, adding that information to your cognitive map. You also have cells that are firing when you reach the border of a certain room, a certain maze, or whatever the case may be. And so your brain is able to put all this information together to form this mental representation. Now, I think we would say that some people do this a little bit better than others, right? Some people tend to be a little bit better at forming maps, and this tends to lead into the gender stereotype conversation about how men tend to do better at parallel parking and reading a GPS and that kind of thing. There are lots of potential reasons why that might be, but men usually do tend to score higher on nonverbal tasks, and women tend to score higher on verbal tasks, and that could be nature, but it could be nurture as well. Anyway. All right, so up until this point, we've been thinking about long-term memory, but there are some other time periods of memory to think about as well. So you see here talking about a sensory buffer or sensory memory. So what happens is that everything in the environment comes in through your sensory memory. So you have information coming in, assuming that you have, fully functioning sensory systems. Everything you see, everything you smell, touch, taste, all of that, everything you hear is coming in and being stored very briefly. So that means that sensory memory has a huge capacity. That's a lot coming in at one time, but a very brief duration. It's only held for a fraction of a second up to maybe a few seconds at most, depending on which type of sense we're talking about. Now, when we're thinking about sensory memory, all of that information that's coming in, not all of that is relevant, right? And if you tried to store everything that was coming in at one time for the rest of your life, you'd be overwhelmed and you'd have a hard time finding what is important. So what happens is that most of that information is lost. But if some of it is deemed important, then it might get moved into your short-term memory. Now, your short-term memory is going to have a smaller capacity. It's not going to be able to store as much. That'll be individual differences, though. Some people have better short-term or working memories than others, and there are certain things you can do to improve your short-term or working memory. And so in addition to having a smaller capacity, it also has a shorter duration. So it talks about lasting up to maybe 30 seconds or throughout rehearsal. When you think about throughout rehearsal, you could technically keep something in your short-term memory forever as long as you kept rehearsing it without ever being distracted. But, of course, that's not really possible. At some point, you're going to fall asleep anyway, right? When I'm talking about rehearsal here, imagine that you wanted to make a phone call, but you couldn't find your phone, and so you were repeating the phone number to yourself over and over again until you found your phone. That would be an example of rehearsal. And as long as you keep repeating that number and nothing distracts you from it, you'll be able to keep that in your rehearsal, in your short-term memory as long as you need to, okay? Now, short-term memory is also known as working memory. The distinction I've always heard here is that working memory is not just holding something, but it's manipulating something, doing something with it. So if I asked you to remember a series of letters in the order that I said them to you, that'd be short-term memory. But if I asked you to remember a series of letters and put them in alphabetical order, that would be working memory. You'd be doing something with that information. But certainly a lot of overlap. So what you can see here, it's talking about the strength of the memory and time. So sensory memory is going to have a high capacity, a lot coming in at one time, but a very brief duration. So most of it will be lost pretty quickly. Short-term or working memory is going to have a smaller capacity. You're not able to hold as many items, but it will have a longer duration than sensory memory, although it's still not super long. Long-term memory is something that has a large capacity over time. You build up a lot of memories and a long duration as well, just like the name suggests. And then intermediate is going to be somewhere in between working and long-term, as you probably guessed, and you'll see in a second. Now, when we think about short-term memory or working memory, it's sometimes discussed in these different components depending on what type of memory it is. So a phonological loop is what we call that component that contains auditory information. So if you're repeating a phone number to yourself over and over again, you would say that's in your phonological loop. The visuospatial sketchpad is holding visual impressions of like remembering what something looks like, maybe remembering the path that you took to get to a certain location. That kind of thing would be a visual memory. And then the episodic buffer is more of an integrated form of the memories, so that you're putting auditory, visual, other components all together, sort of like you're watching a movie, right? So all those would be considered components of short-term or working memory. Now, an intermediate-term memory is something that's longer than a short-term memory, but it's not permanent like a long-term memory. Not that long-term memories are always perfect or always permanent, but they can be permanent. Intermediate memories tend not to be. So, for example, if I asked you, what did you eat for breakfast yesterday? You could probably remember. If you thought about it for a second, you could probably tell me. Now, that's not short-term memory, because I don't know when you're watching this video, but breakfast yesterday was not just a minute ago. So that's not short-term memory, but it's not long-term memory, because you probably are not going to hold that memory in your brain forever unless something really important happened. Like you got engaged while you were eating breakfast, but it's not likely that you'll remember what you had for breakfast yesterday for the rest of your life. Like April 3rd, I'm going to remember breakfast forever. No, probably not. So that's an intermediate term memory. It'll last for a little while, maybe a few days, but it won't last forever. Okay? So we've talked about long-term memory, declarative versus non-declarative, and all of that. We've talked about short-term memory, which can also be called working memory. You could also put another arm out here and include sensory memory that we talked about as well. So with long-term memory, these are going to be memories, just like the name suggests, that last for a significant period of time. Now, long-term memories is not perfect, and we're going to talk about some issues with that coming up. Certainly we can forget things or we can remember them incorrectly, but they do have a high capacity and a high duration. Now, I mentioned earlier about the activity about putting the presidents in, making a list of all the presidents that you know. I do that activity, one, because it tends to be a fun activity. At least people don't have to think very hard. It's a break, you know. But I'm not doing that activity with you guys because it wouldn't really make sense. You just Google the presidents. Anyway, But when I do that in class is not to make people feel bad about the number of presidents they remember, but I do that to demonstrate the serial position effect, which is relevant to long-term memory. So the idea of a serial position effect is that we tend to remember things at the beginning and the end, and we lose things in the middle. So if I called out a list of names to you, you'd probably remember the first few names and the last few names, and you would lose the ones in the middle unless there was a middle name that was very distinctive to you. Like if your name was in the middle, you'd remember your name, but you'd lose most of the rest of them. When I ask about presidents, people usually tell me George Washington, and then they might tell me in the middle something very distinctive, Abraham Lincoln's distinctive, but they lose most of the other ones in the middle. And then they come back and say Donald Trump, Barack Obama, right? So we tend to remember the beginning and the end, and we only remember the middle if it's distinctive. So this tendency to remember the beginning of a list is long-term memory. And it's called the primacy effect. But it's the idea that you've practiced, you've rehearsed it. It's in your long-term memory. Now, if I call out a list of words to you, you'll remember those first few words because you'll have tried. You'll be like, okay, she's going to ask me to write these down. I got to remember, and you'd be repeating them over and over again to yourself. Then somewhere in the middle, you'd say, I can't do it. I can't keep repeating all these. There's too many words. I give up. And then at the end, you'd come back and you'd say, oh yeah, I can remember those last few because the last few items are still in your sensory memory or your short-term memory. And so you tend to do better with the last few items. That's called the recency effect. Okay. This is also true for a lecture or for an in-class lecture too. The idea that you tend to do better at the beginning of class, you're paying attention. And at the end of the class, you bring in your attention, you pay attention again, and maybe you lose some things in the middle. I'm going to suggest that it's a good idea for you with these lecture videos to pause them and take a break and come back if you start to feel yourself losing the information. All right, so this is comparing the serial position effect. You'll see the serial position curve name up here. The serial position effect for two different groups. The control group is in red, and the individuals who have amnesia are in blue. Now, what you'll see is that their memory tends to be worse for all these points, except right up here where they're the same or even a little bit better. That's because this is the recency effect. If I just call the item out to you, the person with amnesia will still remember. Like HM could tell you the last few things he heard, but the beginning items are lost because of that, the amnesia, that loss of long-term memory here. They tend to struggle, but those last few items they can still do. For the typical person, though, you'll remember the first items, you'll get lost a little bit in the middle, and then you'll come back towards the end. Okay, so as I've said, long-term memory is going to have a large capacity and a long duration. It might not feel like this sometimes. Maybe when you're studying for a test, it feels like if you try to shove one more piece of information in your brain, you're going to lose stuff. But we haven't really met the capacity yet for our brain. It has a very large capacity. However, we can make mistakes. We know this. Information can be forgotten or recalled inaccurately. This is going to be something that's going to come up again later in the lecture, but the idea that every time you activate a memory, you take it from long-term memory and you move it into working memory so you can think about it, and perhaps you change that memory. Perhaps the details are a little bit different every time, and then you store that new version. So maybe that fish you caught really did get bigger every time you told the story, that kind of thing. Because when you saved it, you saved that new version that you exaggerated just a little bit on. So if you've told that story over and over and over again, it might not be as accurate as it initially was. This goes into flashbulb memories, right? People talk about, where were you when 9/11 happened? Which, wow. Were you guys alive? Some of you might not have been born yet. Anyway, that's a whole other wow, I'm getting really old. But you ask them, like, where were you now in 20 years? I'll ask you, where were you in 2020 during the coronavirus situation? But anyway, we tend to think that we remember things better when they're emotionally charged, but we tend actually not to do well with that. That's a whole other conversation. Okay, now if we're talking about memory, there are really three tasks that we could mess up at any point. One, we have to get it in, then we have to save it, then we have to retrieve it. So first you have to encode the information. You have to get the information from the environment into your short-term memory. It's got to pay attention to it, get it in there. Once you get it in there, then you need to consolidate it and save it into your long-term memory storage, and then you need to be able to retrieve it when you need it. So this is like listening to the professor explain something, and then studying it, and then answering a question about it on the test, right? Which your activity for this week, for those of you who do the activity, asks you about study tips based off of this chapter, and we'll talk more coming up, but how should you be studying for tests based on the information in this particular chapter? That might not be as much fun as the president's thing, but I felt like I couldn't really do the president's thing outside of class. So there you go. All right, so what you have here is just sort of a picture of, it's the Atkinson and Shiffrin model, but this model with sensory memory, short-term memory, and long-term memory. So you have information coming in from the environment, and that's going to come into your sensory buffers or into your sensory memory. Most of that will be lost, but whatever you pay attention to gets encoded, gets into your short-term memory. Then, if you continue to rehearse it, you can keep it there for a while. Maybe you use it and lose it, that kind of thing. If you're able to consolidate it, you move it into long-term memory, where it stays until you need to retrieve it. You bring it back into your working memory so you can use it again. Okay. Actually, what I'm going to do at this point, I can't make you, obviously. I'm going to recommend you take a break. This is a long lecture. I don't know, maybe halfway through. This is where I would normally have stopped the lecture, and then we would have done this in the next class period. So I'm going to take a break. Of course, I'm going to cut out my break, so it's going to be just a second for you guys. But pause the video, take a break, get a snack, get a Coke, and come back, okay? Alright, so I hope you guys took a break. I took a break. And one other thing to say is that my kids are outside playing in the yard with my husband. So if you hear background noise, that's what's coming from that. The front yard is the only trip they can take at the moment. So anyway, alright, so carrying on talking about memory, we talked about how you have to get the information into your memory, you have to encode it, and then once you do that, we need to consolidate it, we need to save it, and then you need to be able to retrieve it. Now, one question you might have then is, where is this information consolidated? Where is this information stored? So that's going to be something that we're going to touch on throughout the rest of these slides. But one thing to think about is that this is an ongoing area of research. We don't know everything about this, and most of our research does come from case studies with individuals who've had some type of amnesia. So bearing all that in mind, starting off with encoding an fMRI, so looking at what parts of the brain are active during particular processes. When they have individuals in an fMRI and ask them to recall spatial information or pictures, we see that structures on the right side of the brain tend to be a little bit more active, especially the right prefrontal cortex. And then there's also going to be some activation on both sides as well. It talks about the parahippocampal cortex. That's going to be the cortex that's near the hippocampus. But for the prefrontal cortex, the right side is especially important for spatial information. Then for verbal information, we're going to see that the left side of the brain is going to be more involved here. It talks about the left prefrontal cortex, the left parahippocampal cortex as well. This is going to be consistent with what we're going to talk about in chapter 19. When we talk about language and lateralization, we'll see that for most people, not for all people, but for most people, the left side of the brain has an advantage with verbal tasks, and the right side of the brain is more involved in spatial reasoning. Alright, now we encode information, we get it into our memory, and then it needs to be consolidated, it needs to be stored. This slide talks about an engram or a memory trace. This is just referring to a physical change that happens after you've had some type of learning experience. And we're going to talk a little bit more about this in the upcoming slides, so hang on for that. But the question is, where do we have permanent storage? Where do we consolidate this information? Well, one thing that we know from HM is that it's not in the hippocampus. Okay? HM was able to keep the memories he had already stored before his surgery. He retained those memories before, and he had his hippocampus removed and other large portions of his brain. So then the question is, where is it stored? If the hippocampus is helping us make new memories, then where are they stored? And what we're going to see is that it tends to occur in the regions where it was first processed. For example, the occipital lobe is important for visual information, vision. We skipped the chapter on vision, but we mentioned occipital lobe earlier in the semester. That's in the back of the brain, important for vision. And perhaps that is where the memory, the final consolidated version of the memory, is stored when it's a visual memory. So it seems like whatever part of the cortex was used to process the information is also where that information is housed. So as long as that part of the brain is not damaged, you would be able to retain that memory. Now, we talked about encoding, getting information into your memory. Consolidation, we're going to save it, and then we retrieve it. We bring it back into our short-term or our working memory so that we can use it. The question then is, if you take information from long-term memory and move it into short-term or working memory, does that make it plastic? In other words, does that mean that you can update that memory, change it, and then reconsolidate it or resave it in that new updated form? There does seem to be some evidence to support this. I think we're all aware that this can happen, that our memories are not perfect, and that if we tell a story over and over again, maybe it changes a little bit every time until eventually we're not 100% sure what our initial experience was like. We know our memory is not perfect, okay? Now, what does this mean for eyewitness testimony? There's a lot that could be said here, and I won't go into any great detail here. If you take a memory class or other classes, you'll probably have that conversation. The thought then is eyewitness testimony is often very powerful. It's very convincing when a victim points at a person and says that was the person who, whatever the crime might be. However, we know that our eyewitness testimony is not perfect. There are plenty of documentaries that you can watch of situations where a person was not lying, the victim wasn't lying, they were convinced that this was the right person, but then something like DNA came along and proved that that was not the right person. So there's nothing we can do to cause our memory to be perfect. There are certain ways that you can interview someone to try to reduce this problem. So it would be better if the police officers, whoever's doing the interviewing, did that very soon after the event and did that alone with that person or not with other people that might cause them to feel pressured about not saying this or that. Asking them open-ended questions, not leading them. Sometimes we can accidentally or intentionally lead with our questions like, and then they did this, didn't they? That kind of thing. But instead just asking like, what happened next sort of questions. There are ways that you can interview a person that can help prevent issues with eyewitness testimony, but we can't stop all of those issues, okay? So just being aware that your memory may not be as accurate as you think it is, which is sometimes scary. All right, now, I mentioned in chapter 16 just a tiny bit about PTSD, and I said that we would talk about it more in this chapter. So PTSD, post-traumatic stress disorder, is a situation where a person has undergone trauma and then they have lingering effects of that. There are lots of different symptoms you can see with PTSD, but many of them are memory related. So you see things like nightmares about the event, flashbacks to the event, almost obsessions, like having a hard time stopping, recalling the event. And so then the question is, why is that? Well, the thought is that when you're in a stressful situation, you have stress hormones that are released, just like we talked about with chapter 15, and that seems to strengthen that memory to a certain extent. It makes that memory more powerful for you. Not always more accurate, but more powerful. Harder to stop thinking about it, harder to distract yourself from it. So it talks about a few different things here, GABA, opioid, but especially epinephrine and norepinephrine. It seems that, at least in animal models, and this holds true for humans as well, is that when you have high levels of epinephrine and norepinephrine in your system, then you remember an event more intensely. And we know that if a person is in a traumatic situation, they probably will have high levels of epinephrine and norepinephrine. So there are lots of different treatments for PTSD. The prevailing behavioral treatment, cognitive behavioral treatment, is exposure therapy, which sounds awful, but it's really effective. Having the person not avoid the incident, but talking about it. So the behavioral part is exposing the individual to it, talking about it, and keeping them from avoiding it. But then the cognitive part is also trying to change the way they think about the situation. They might feel like it was their fault, or they might feel like they have no control over the situation, that kind of thing. Just changing the way they think about it so that it doesn't seem as negative for them, okay? Now, could we combine that cognitive behavioral treatment with the biological treatment as well? So, it talks here about blocking chemicals that act on the amygdala. We know the amygdala is the part of the brain that's involved in negative emotions, especially fear. So, if we can impact the amygdala, then perhaps we can help with some of these symptoms. So, the research on this was done with propranolol, which when I first saw this, almost startled me. I used to take propranolol. It's a blood pressure medication, or at least I was taking it as a blood pressure medication. And I can remember when I first started taking propranolol that it did make me feel very tired, very sleepy, kind of calm, almost like not quite benzodiazepine level, but it certainly made me feel very tired and calm. One thing about propranolol is that in addition to lowering blood pressure, it can also decrease excessive heart rate. Your heart rate's too high. OCD, type A individual, I struggle with that. So anyway, that's a whole nother conversation. But what they did was they took individuals who had symptoms of PTSD and they treated them with propranolol. Now, what they would do is they would have them write down an account of their story, and then they would have them read that account while they were under the influence of the propranolol. And they did that multiple times. The idea is that it's not going to stop them from remembering the event, but perhaps if they're taking a substance that calms their body down and reduces epinephrine and norepinephrine while they're reading their story, that will make it seem less upsetting to them, less intense. And then they could save that new, less intense version in their brain as opposed to the previous version, and that this is something that could be really, really helpful. Now, the research on this, how we knew that this could potentially be helpful, is that they brought in individuals and they had some people who were in the control group, so they did not receive the medication, some people who were in the experimental group who did. So then they had both groups read emotionally arousing stories, so maybe something that's scary or a little disturbing, and then emotionally neutral stories, so, I don't know, just a boring story. And what they found was that for both groups, they remembered the story. But what happened was that the controls remembered the emotionally arousing story better. So there's something about the propranolol that not that makes you forget the story, but maybe the story doesn't strike you as being as important. It doesn't stand out to you as much. It's easier for you to repress some of that and just say, oh, that's not important. So there was something about reading a disturbing story while you were taking propranolol that caused it to be a little less salient for you. It just didn't stand out to you as much. And so because of that, we started getting the idea that this could be helpful for people with PTSD. All right, so when we're talking about memory, we're talking about having some type of learning experience, and then you're able to store that information relatively permanently in your brain. So that requires neuroplasticity, which is a concept we've mentioned a few times throughout the class. The idea that neurons and neural circuits, so groups of neurons that are working together, can be changed in response to a learning experience, something in the environment. If neuroplasticity was not an option, then we couldn't learn anything new. We couldn't save any new memories. So we should definitely be thankful for that. Now, we did talk earlier in our developmental chapter about how neuroplasticity is at its height during those beginning stages of development. But even adults continue to have some neuroplasticity as well. So we're going to talk more about neurogenesis in a little bit, about creating new neurons, and we'll see that that does continue to happen to a certain extent, a limited extent, in adulthood as well. So all of this is allowing us to continue to learn and grow throughout our life. Now, I'm not going to go into as much detail on this topic as your textbook does, okay? But let me know if you have any questions about any of this. We're trying to understand that engram that I mentioned earlier, that physical trace that's left behind when learning happens, something that we could actually see. So if we're looking under a microscope, what will we see when an individual learns something? Now, Sherrington is the researcher that is mentioning here, talking about the fact that perhaps it's changes in the synapse that allow for learning. So it's been a little while since I've seen you guys in class. Certainly been a while since I drew my terribly drawn picture of neurotransmitters and the synapse and all that up on the board. But remember, you have your presynaptic membrane, right? So you have the end of this first neuron, okay? You have the axon terminal, and then you're going to have the neurotransmitters being released from the axon terminal. They're going to diffuse across the synapse and bind to receptors on the next neuron, which could be on the dendrites or on the cell body itself, and that the second neuron is the postsynaptic membrane, right? Okay. So what we're saying is that you could have changes in the presynaptic membrane. So we could have some type of change that causes the neuron to release more neurotransmitter or less neurotransmitter. We could have a postsynaptic change, so we could have a change in the number of receptors, for example. We talked about upregulating and downregulating receptors and all of that. So because of that, Sherrington would speculate that when you learn something new, you have some type of learning experience, then your synapses change a little bit. Now there are a few pictures coming up to kind of show you what I'm talking about here. So you see the axon terminal, so this is the presynaptic membrane. We've got the neurotransmitter here, and then the postsynaptic membrane here specifically is the dendrite. So it says PSP over here, okay? We're talking about the postsynaptic potential. So remember that when you have neurotransmitters come across and bind to receptors, that changes the postsynaptic potential here of the postsynaptic neuron, which means that if you're having an excitatory postsynaptic potential, that makes this neuron more likely to fire. If you have an inhibitory postsynaptic potential, this makes this neuron less likely to fire. Okay, so what we're saying is this is what's happening naturally before learning takes place. And then after training, you might see a few different changes. One, you might see more neurotransmitter. You'll notice that there's less neurotransmitter before, more neurotransmitter after. Or you might have the same amount of neurotransmitter you initially had, but you might have a growth in this dendritic spine. You might have more receptors here. Or it could be that you have both. You have an increase in neurotransmitter and an increase in receptors. So all of these things could be working to increase the postsynaptic potential. So we have a physical change in the synapses because learning took place. Okay. It also talks about changes in the rate of inactivation of transmitter. So remember that there are some neurotransmitters that are taken back up whole, right? And so we talked about like with selective serotonin reuptake inhibitors, blocking that. But some neurotransmitters are broken down. We talked about like acetylcholine esterase that breaks down acetylcholine. So if a learning event changed the rate of inactivation of a neurotransmitter and cause the neurotransmitter to be in the synapse longer and be active longer, then that would cause an increase in postsynaptic potential as well. It also talks about inputs from other neurons. So I have a picture here that may make this a little bit easier to see, right? But what can happen is that before training, you have this neuron talking to this neuron, okay? But now we've trained the individual, we've had some type of learning experience, and now this neuron is talking to this neuron, and this neuron is talking to this neuron. And because of the increased input here to this neuron, it's releasing more neurotransmitter, which is then going to have more of an effect on this postsynaptic membrane. So we have that increased postsynaptic potential. So what we're essentially saying here is you can form new connections after a learning experience that then cause an increase in firing if it's an excitatory postsynaptic potential. So what we're saying is that these synapses may provide long-term storage. We have long-term changes in the synapses, okay? New synapses could form, just like the one we saw on the previous slide where we had a new connection with this neighboring neuron. Or it could be that there's a connection that's not being used, and training could prune that. So we're not just forming new connections, but in addition, we have to get rid of the connections we're not using so that we can put all of our energy and resources towards the connections that we need. Okay? So if you look at what's happening here, okay, before training, we have these neurons talking to each other. This neuron's not really doing a whole lot at this point, so these are not really connecting with each other much. But this neuron is communicating with this neuron, and here we have a new neuron, maybe that's starting to grow. We have a dendritic spine that might be sprouting here. And after training, we have a new connection that formed, okay? This neuron was very active previously. It's been stimulating this neuron. And so now because of that, we've had some type of learning experience, and we have a new dendritic spine pop up here, and we have a new synapse that's formed. So that would be adding new synapses, new connections. But also, we might prune some unnecessary connections here, and that would be helpful for us because we want to put our resources towards the connections that are most active. So what we're saying in this particular picture is that we have this neuron being active and communicating with this neuron. So this is a healthy connection. Here, there's not a whole lot going on. Well, because this neuron is so strong and so reactive, after training, that could cause this neuron to start to communicate with this one, the next to it, and it could prune this connection that wasn't being used. So once again, putting our resources towards the connections that are needed, the connections that are actually working, and pruning the unnecessary connections, which is a process that continues throughout life. All right, now, some research has been done looking not as much at the cellular level, but a little bit at a larger structural level here for lab animals in different environments. And this research was done with mice. I think there's a picture. Yeah, there's a picture here that you'll see in a second. This research was done with lab mice, and they were interested to see if I put you in a particular condition, will that then impact your brain structures? Will that cause a difference in your the development of your nervous system? So there were some mice that they were looking at that were in standard conditions. So you'll see the standard condition involved a small, relatively boring cage, but at least you have a friend, right? You also have some of these mice that were in the impoverished condition. So just like it sounds, they're not going to have as much stimulation. Here what's happening is they're in the simple, boring cage, but they're by themselves. And then finally, the enriched condition is going to have lots of different things for the mice to do, and lots of friends for them there, okay? And maybe perhaps even more food available, that kind of thing. All right, now, what's happening is that we're randomly assigning mice into different conditions so that the only difference between them is the type of condition they're in, and we're interested to see if the mice in the enriched condition will have better outcomes compared to the mice in the standard or the impoverished conditions. Okay, so here's what they found, and this is comparing the enriched condition with the impoverished condition. So you can see that this poor mouse is not having the same experience as his friends down here. Okay, what they found is that animals who were in the enriched condition, so they had things to do, they had playmates, etc., they had heavier, thicker cortex, so there's more brain matter there. Enhanced cholinergic activity in particular, so the neurons that are producing acetylcholine are especially active, and you're going to see this mentioned later, but increasing the effect of acetylcholine is thought to help with learning and memory, so perhaps they had better ability to learn from training experiences, that kind of thing, and more dendritic branches, more dendritic spines, more synapses. So the thought is that when they had learning experiences, when there was something enriching going on in their environment, that caused them to have more brain matter, more connections, more acetylcholine, all of that, compared with our poor mouse in the impoverished condition that didn't have that going on. Now, I don't think it's too much of a stretch to say here, then let's think about what we're doing for ourselves and for our children. Let's make sure that, you think about kids, make sure they're in an environment where they are loved, cared for, and given structure and books, and I put books really high up on my list of things kids need. Not above love, but pretty close. Okay, having said all of that, think about yourself. Maybe you feel like this poor mouse right now, right? Can anybody relate to this? Maybe you're quarantined and you're stressed out and you feel like this mouse. I'm sorry. Yeah, I miss you guys. But I hope you're doing well. But the idea here is that you need to find ways to be these mice. Find ways, safe ways. You know, have some Zoom video calls or, I don't know. Take care. Just take care, okay? I mean that in the best possible way. Alright, so now if we're studying changes that are happening when an individual learns something new, it might be beneficial to try to study this in very simple creatures. And that's what they've done here. Aplysia, I don't know that that's the right pronunciation. That's what I'm going to go with. They're talking about a sea slug here, essentially. And when they're talking about this particular creature, it's a very simple creature. So it talks about having fewer nerve cells. There's not that much going on. There's not as many neurons. And because there's not as many neurons, and because they're very simple creatures, there's very little variation between them, so that whatever you find with one particular little sea slug, it's probably going to generalize to other sea slugs as well. And because there's very little difference, we can use that to come up with pretty detailed maps, because the circuits are not terribly complex compared with other creatures as well. So we're talking about our little Aplysia, our little sea slug here. I don't know if there's a picture of the sea slug. Perhaps I should have left that in here. But anyway, I think you guys have the general idea of what I'm talking about. So, because of that, there's some advantages to working with very simple creatures, although that might not tell us as much as studying more complex creatures. We can learn more in depth with simpler creatures. So, invertebrates demonstrate non-associative learning. So, when you have a very, very simple nervous system, you're not going to have as many abilities as an individual with a more complex nervous system. But even here with these very simple nervous systems, you can still see this non-associative learning. Non-associative learning basically involves a single stimulus presented once or repeated, and there's a change in something about their response because they've been exposed to this stimulus before. Okay. Now, these that are listed here as examples, these are things that happen in sea slugs, but they also happen in us as well. Okay. First of all, habituation, a decreased response to repeated presentations of a stimulus. This has probably happened to you. Maybe you stop hearing the clock ticking on the wall. Or maybe you stop smelling whatever the smell is in the environment. You habituate to it. So it's some type of stimulus that's repeatedly there, and your brain sort of blocks that out. So that's habituation. And we'll talk about an example of habituation in our sea slug friend in a second. But you can also have dishabituation. You can have that response restored. Okay? So maybe you don't hear the ticking on the clock until there's some other loud noise that scares you, startles you, and then after that you hear the clock again. That's dishabituation, which means you stopped being habituated to it. Now you can hear it again because there was some type of stimulus that startled you, scared you, or just brought your attention back to the current situation. Sensitization happens when habituation has not happened. So with dishabituation, you start to notice a stimulus again that you had previously not been noticing. With sensitization, you're still noticing that stimulus, but now that stimulus seems even stronger, perhaps. Like I was still hearing the noises around me, but then something happened that scared me, and now I'm hearing it even more loudly, perhaps, okay? So these are things that happen to us without us necessarily paying a whole lot of attention to it, but they also happen to simple invertebrates. How does this happen in our sea slug that we've been talking about? Well, remember that habituation happens when the stimulus happens over and over again, and there's some type of change because of that. Like you've been hearing that clock ticking, and then eventually, because you've been exposed to it so much, you stop hearing it. Well, something that they'll do with the sea slugs is that they'll squirt water on its siphon. So its siphon is a tube. If you have a picture, if you look in your textbook, there's a picture of a sea slug. It's just a tube that the sea slug has that it uses to draw in water, And when you squirt water on its siphon, it scares the sea slug. Essentially what's happening is that it retracts its gills because it's scared. It stops and panics a little bit. However, if you continue to squirt water on the gills, the animal will gradually over time retract the gills less and less. Essentially, it will say, okay, well, you've been squirting me with water for a while now, and it hasn't damaged my gills at all, so I guess this is not a threat. Okay, so this is habituation. Initially, it was responding to the water that was being squirted on it, but after enough exposure to it, it stopped responding to it. Now, habituation is a type of learning. You have an experience with something in the environment, and you have some type of change because of that. What type of change is happening? Well, it's talking about the habituation causing synaptic changes between the sensory cell in the siphon and the motor neuron that retracts the gills. Okay, so there are sensory cells in the siphon that detect the water being sprayed on it. Initially, this startles the sea slug, and a message is sent from the sensory cells to the motor neurons. We need to retract the gills immediately. But then, as you continue to produce that sensation, over time, learning takes place, and this circuit becomes less active. Less neurotransmitter is released, and therefore you don't have as much of a retraction from the motor neurons. So this is all just saying that learning seems to involve changes in the synapse, not just in more complex structures, more complex individuals, but in very simple invertebrates as well. Now, as I said, there are reasons why this type of research is easier in creatures that have simple nervous systems. Mammals tend to have pretty complex nervous systems, but there have been some circuits related to learning that have been identified in mammals as well. One that your textbook gives as an example is this eye blink reflex. Okay, I have an eye thing. I don't know if I've mentioned this in class before. I wear glasses because I cannot wear contacts. Like the idea of poking myself in the eye. My husband wears contacts, and I can't even watch him putting in his contacts or taking out his contacts, okay? And I don't like going to the eye doctor, in part because I don't like it when they puff the air in your eyeball, right? Who likes that? Nobody likes that, okay? But what we're saying here is that an air puff blown into your eye is an unconditioned stimulus that will cause an unconditioned response of blinking. You don't have to learn that. That is a natural, normal reflex to protect yourself. But what happens if we bring in a previously neutral thing, like this acoustic tone, a noise? We play the sound and we puff you with the air. We play the sound, we puff you with the air. Initially, the noise was neutral, but now, because it's been associated with the air puff, it has become a conditioned stimulus. You've been conditioned to respond to it. And now you'll have that conditioned response of blinking even if you only have the noise without the air puff. Okay? So the unconditioned stimulus of the air puff causes the unconditioned response of blinking. Then we pair the air puff with the noise enough times over and over again. The noise becomes a conditioned stimulus capable of producing the conditioned response of blinking. You'll notice that the unconditioned response and the conditioned response are both blinking. The difference is that the unconditioned response happened naturally to the air puff. The conditioned response happened to the noise, which you had to learn that. Okay. Now, this particular circuit is thought to be, in part, happening in the cerebellum, so that if an individual has some type of damage or some type of medication that's impacting the cerebellum, then that can impact classical conditioning as well. So one other thing that your textbook says about this process is that GABA antagonists cause conditioned behaviors to disappear, at least until the drug wears off. So what we're saying is that if you train an individual to blink to a puff of air, and then you give them a GABA antagonist, so that's something that's going to block the effects of GABA, then at least for the period of time that they're under the influence of that drug, they'll lose that behavior they had learned. They won't blink in response to the noise. So this is taken to mean that likely something happening in the cerebellum that involves GABA is important for at least classical conditioning in mammals. That's the thought. Okay, Now, a little bit more about synaptic plasticity here, about ways that the synapses change when we have some type of learning experience. So you'll see that this talks about a researcher. His name was Donald Hebb. So when you see that this was about him, about Hebb's ideas, that's where we come up with the term Hebbian synapses. Okay. So Hebb proposed that when two neurons are activated together, their synaptic connection becomes stronger. Essentially what he's saying is that the neurons that fire together wire together. So if you have some type of experience that causes these two neurons to be activated at the same time, then their connections will become stronger and stronger. So we're saying that something in the environment has caused these neurons to fire, and then the result of them firing at the same time is that they now are stronger, and that connection has become stronger. This would make sense because connections that are weak, that are not being used, are going to be pruned, but the connections that are being used are going to receive more of the resources, right? So when it talks about cell assemblies, these are groups of neurons and that are together. They're linked via Hebbian synapses, so they're firing at the same time. We're saying there's a group of neurons that are firing at the same time, and the thought is that this could be related to memory storage, okay? Now, one thing that's interesting here, it talks about tetanus. We're not talking about a rusty nail, need to go get your tetanus shot. Here, in this particular example, we're talking about brief high-frequency burst of electrical stimuli. So what they would do is that they would apply this burst of electrical stimuli. That's going to cause the neurons to fire, right? We're forcing the neurons to fire. We have a very strong, powerful stimulus coming in that causes the neurons to fire. Now, what we found is that after that stopped, that's not happening anymore, the neurons are still firing more strongly. It's as if when you had the stimulus that was causing the neurons to fire frequently, they were wired together, they were connected together, that connection became stronger, so that even after you stop applying that stimulus, they're still more powerful than they were initially. And the thought is that this could be related to memory, okay? So the concept behind this is long-term potentiation. You see this LTP acronym here. A stable and enduring increase in the effectiveness of synapses. You have a learning experience that causes the unnecessary connections to be pruned, and the necessary connections, the neurons that are firing together at the same time, will become more effective. They will be more powerful. And because of that, now we have a synaptic change as the result of learning something new. So the thought is that long-term potentiation could be proof that memory has taken place, proof that learning has taken place, that memory is being stored, okay? So then the question is, if we're talking about long-term potentiation as if it is memory, are they the same thing, right? Now, if you have any questions about this, let me know. I just want you to kind of understand the big picture idea here. You don't have to know all the details that are in your textbook. I'd focus on the specific sections of your textbook that we talk about in the slides, and email me if you have any questions here. We don't know for sure that long-term potentiation causes memory to happen, but there is some evidence to suggest that it seems likely. One thing is correlational observations. Of course, correlation never equal causation, but what we're saying here is that the time period is pretty similar. So with long-term potentiation, this is something that can happen very quickly, but then can last for weeks or more, which would mean then that it could have a similar time course as memory. There are memories that last for weeks or more, perhaps. Also, somatic interventions. So remember that with a somatic intervention, you're changing something about the body, changing something biological, to then see if you cause a change in something behavioral. So what they did is that they have certain drugs that they can administer that block this process. For example, glutamate is important for this LTP process to take place. So if they block glutamate, then that can prevent LTP from happening, and that also impairs learning. So we block long-term potentiation, and then because of, well, I don't know if I should say because of that, but immediately following that, you have a impairment in learning. So it seems like they're definitely related. And then also with the behavioral intervention, so this is an experiment where you're changing something in the environment to see if you can change something biological. When you train an animal, that can cause long-term potentiation to happen. So certainly there's evidence to suggest that there's a close connection here, although I'm sure more research will continue to be done on that. Okay. Last couple things to talk about in this video, adult neurogenesis. So we're talking about the production of new neurons. Now, the majority of that is happening earlier in life, but you do continue to form new neurons to a certain extent throughout your adult years as well. It talks about this occurring primarily in the dentate gyrus, which is related to the hippocampus. We talked about how hippocampus is important for memory and learning. So these new neurons that are coming up in the area of the hippocampus are more plastic, which means they're more influenced by the environment than neurons that were there before, and show enhanced long-term potentiation, which might mean that these neurons that are coming up later in life are serving the purpose of helping you continue to learn new things. That would make sense. Now, when it talks about conditional knockout studies, this is research that's been done in lab animals where they turn off neurogenesis. So they stop the animal from producing new neurons in adulthood, and what they found was that these individuals, these lab animals, struggled with spatial learning tasks. So it's thought that there are some memory tasks that are enhanced by these new neurons that are formed in adulthood. Some of them may not be, but some of them are. Now, remember that the dentate gyrus is close to related to the hippocampus, and so probably memory tasks that involve the hippocampus would be especially harmed by this conditional knockout because we're preventing these new neurons from being produced in that area. It also talks about how you can have the olfactory bulb continuing to produce new neurons. Just like it sounds, olfactory bulb being important for smell, I think we would say that smell can certainly be strongly associated with memories. Maybe there's certain smells that automatically trigger certain memories in you. Okay. Last real topic here is about age-related memory impairments, what happens as we get older. We did talk a little bit about this in the developmental chapter where we talked about Alzheimer's disease, but just to touch on a few things here, there are some aspects of memory that individuals tend to have problems with as they get older, not necessarily every aspect of memory. It talks here about impairment of coding or encoding and retrieval, so getting information into your memory and retrieving information from your memory. This seems to be limited to specific situations where you're trying to remember something. Your textbook talks about it being a self-initiated process. If you put up a picture of someone they knew, then they didn't have trouble remembering that face. But if you were asking them to initiate that memory themselves, they didn't have the picture in front of them, then that made it a little bit harder for them. So there's certain aspects of getting information in and retrieving information that seem to be less powerful as you get older. It talks about loss of neurons and neural connections. So I think I mentioned in class that your brain starts losing weight after you turn 30, which might be more disturbing for me than for you guys. But anyway, the idea is that you start to lose some neurons, and especially certain neurons that may be important for memory. So we've mentioned a couple of times about acetylcholine and how that's important for learning and memory, and related to Alzheimer's disease as well. So you'll see this here. There we go. It talks about deterioration of cholinergic pathways. You don't need to know, it talks about the septal complex and the nucleus basalis of Maynard. Don't worry about that. What I want you to get here is that the pathways that are providing acetylcholine to the hippocampus and the cortex can start to deteriorate. That is normal to a certain extent. That happens as people get older, but what we see is that individuals who have Alzheimer's disease have much more severe loss here. And the thought is that the loss of the acetylcholine could be could be a cause behind Alzheimer's. So then the question you might ask yourself is, how can we treat that? We don't have any cures for Alzheimer's right now. I really hope that we do someday. I had a grandfather that passed away from Alzheimer's. I know how cruel that particular disease can be. One thought then is that we need to get more acetylcholine into the nervous systems of these individuals. And how are we going to do that? Well, it talks about nootropics here as a class of drugs that enhance cognitive function. I think that this would be something that would fly off the shelves, right? If we could come up with a drug that enhances cognitive function, that helps with memory, that helps with attention, that was safe and didn't have side effects, I think that that's hard to imagine that happening, but I think if we could have something like that, I think it would be very popular. But some options that people are working on, it talks about cholinesterase inhibitors. So acetylcholinesterase is the enzyme that breaks down acetylcholine, so that if you block that enzyme, then you can have acetylcholine in your system longer, and that tends to show some benefits for earlier stages of Alzheimer's. It's not a cure. It may be something that can hold off the symptoms for a little bit in a more mild, moderate stage of the disease. But then as it progresses, then you're going to lose that benefit. It talks about ampakines as well. So I mentioned earlier that glutamate was important for long-term potentiation. And so if we could come up with a drug that would increase long-term potentiation, then that might help treat memory issues, help individuals to learn and continue to maintain their memories a little bit better. Last thing here, think about the mice in the enriched conditions. We want to be that mouse. So lifestyle factors, living in a favorable environment, what does that mean? Wow, we tend to be living in a limited environment at the moment. I understand that. One of the things here is having an above average education, which I hope you guys feel like you're receiving that. Also, engaging in cognitive tasks, doing something, using your brain, I see that mentioned here, complex and intellectually stimulating activities. Do something new, read a new book, do some Sudoku, that kind of thing. But then also, there's research to suggest that having intact families, good family relationships, is also associated with a reduction in cognitive decline. Being active, exercising, taking care of yourself, sleep is a good idea here. It talks about having a partner of a high cognitive status. I would say this is correlational, but it makes sense. You can't randomly assign someone into who they're going to partner with, but I think it does make sense that having someone who's there with you that you could have stimulating conversations with could definitely be helpful. Okay, so I hope I didn't lose you guys there. Let me know if you have any questions. You do have an activity this week that's due by Friday at 5 p.m. It's just asking you to think about how you could help with your memory with things like studying. So you want to think about spacing out your studying, not trying to cram all at one time, making sure that you're resting, getting plenty of sleep before you're taking a test, that kind of thing. So complete that activity if you would like to or if you need to. Remember, you do get 2 drop grades for activities. Let me know if you have any questions, and I will talk to you guys during next week's video.